Dear students, now I'll be elaborating on the types of FASTA or fast alignment algorithms and the types that are available for the algorithm. So to start with, the fast alignment can help you search and compare the protein, DNA or RNA sequences by looking at the database for each one of them. Also, once you have found the matching words within the query sequence, then you can use these words and perform the Smith-Waterman algorithm and arrive at an overall alignment. So to start with, the first type of FASTA is the FASTA35. So it can help you to scan a protein or a DNA versus another protein or DNA database. So essentially, any comparison that you want to do can be performed by FASTA35. The next type of FASTA is the FASTX35. So this can help you to compare the translated DNA sequence to a protein sequence database. So on one side you have a DNA sequence, that is your query, and on the other side you have a protein database. So all 6 open reading frames are calculated by looking at the DNA or the RNA sequence, and then they are converted into a protein for comparison. The third type is the TFASTX35. So it can help you to compare a protein sequence to a DNA database. So on this side, that is your query is a protein sequence, while on the database side you have a DNA database. So what you do is you take one sequence from the nucleotide database and you compute all 6 ORFs and translate them into proteins and then perform the comparison. Next is the FASTY. So it can compare a DNA sequence, that is all 6 ORFs, the open reading frames, and you can compare them to the proteins that are there in the protein database. Next is the FASTS35. So this is a special type of FASTA because it can help you to compare unordered peptides to a protein sequence database. So if you have some unordered peptide, then you can look at the database as well, and this can be extremely useful and powerful tool as well. The next is the FASTM35, wherein you have an ordered set of peptides versus a DNA database. So when I say a DNA or an RNA database, so essentially it's a nucleotide database, and we compare the all 6 open reading frames that can be created by each sequence within the database, and then look at the protein sequence followed by its comparison with the with your query sequence. So in conclusion, there are several types of FASTA which you can use depending on your need, and that these algorithms can help you to create alignments and comparisons between sequences in a very fast manner. Also, several types of these algorithms are there and all of them are available online. So you don't have to write them yourself. All you have to do is just go to the corresponding online resource and utilize the algorithm.